Good afternoon, guys. I know we didn't have time to do the photosynthesis lab before we were sent home, so I'm going to try and do a couple of videos here to help you with the photosynthesis lab and show you what we would have done in class. But we're not in the classroom, we're in my house, in my kitchen. So welcome to Kitchen Science Lab. The first thing we're going to do today is the paper chromatography, and with the paper chromatography, we're trying to see what colors of pigments are actually in our leaves. So the leaves that we can use are just spinach from the grocery store, and the materials that we can use at home that are very similar to what we would do in the lab are just a regular old coffee filter, a spinach leaf, a coin, and I'm going to try two different types of solvent to see if I can get the chlorophyll pigments to separate out on our chromatography paper. The first one I have is rubbing alcohol, and the second solvent that I'm going to try is just a fingernail polish remover that is made of methyl acetate. I know acetone would work, so if you want to try this at home and you actually have some fingernail polish remover made of acetone, you're welcome to try that. So the first step in this experiment is to take our coffee filter and to cut it into just strips. And this is actually the same shape of the strip that is in your lab manual. You need to have a glass or something to set the paper strip in so that just the tip is touching down at the bottom. And so I have two of these little setups with juice glasses. I just, again, cut my coffee filter. So the way that we're going to transfer the pigments in this leaf to this piece of paper is with the coin. So bear with me, this is my iPhone. So I'm just going to line it up here, like such. And I'm going to take the coin, trying to prop up the phone here. You can see the phone. Just going to take the coin and rub it up against that leaf several times, because what you're trying to do is you're trying to get a green line on there. So let me go ahead and do that a few more times and not drop the phone. See, if I scoot this back here, you can see just out of sight. And you want to try and get as much of that green pigment on the filter paper as possible. So filter paper, coffee filter. In the lab we would have used a coffee filter. So even just rubbing it or squishing it down on there, pretty good. And so I'm going to try two of these. Whichever solvent works better, whichever one you have at home, you can certainly try that. Smush it on there a few times. If you only have the brown coffee filters, it may be harder to see, but you can see we got pretty good green lines on our filter papers. We want to let them dry a little bit, make them dance around. And then we're going to sit them, make sure that they kind of balance in our little cups. I'm going to write on our cups here that this is going to be the alcohol and that this is our nail polish remover, so we can maybe figure out, hypothesis right here, which one of these is better. So I'm going to set this here, stagger them a little bit. That would be up a little bit higher to mark our rolls. Okay, so I'm going to take this out of the way so I don't splash. Stuff is worth its weight in gold right now, right? Rubbing alcohol. Okay, I'm going to just balance that in there. So just the tip, if you can tell in the cup, and you can, that just the tip is sitting down there in the in the alcohol. And we're going to do the same thing over here on our alcohol, or on our nail polish remover. Also worth its weight in gold. And so just the tip, and immediately the liquids are starting to come up. The reason they're doing that is through capillary action, that the pores on the paper, all those little filters overlapping, leave lots of spaces for our solvents to be traveling up. And so they're traveling up through the filter paper, and they're going to interact with our pigment molecules, and we'll see if anything happens as a result. And what we should see happening, hopefully we do, and if we don't, I'm going to just do a little mock-up with some markers, because it also works with ink, is that the pigments that are lighter weight are going to travel further up through that paper than what you would see with the heavier pigments. The heavier pigments have a harder time navigating and moving up through the filter paper, and the lighter this smaller size molecule, it can weasel its way up and travel up the filter paper further. Can't really see much happening here. It does look like our nail polish remover, if it's having some effect, and the alcohol has not yet traveled up to where our pigments are. So, but you can see, kind of, if I zoom in here on this one, the other one with the nail polish remover, we do see it traveling up, and we do see the pigments starting to separate out. Put this back up. All right. The alcohol, it just seemed to stop. It's not really moving. So maybe we'll just put that one out of view. Oops. So we're here to see the spinach pigments migrate. So what we would do is we would let these liquids, the solvent, migrate until it's near the top of the piece of paper, so almost at the top, and then we're going to record. So now that you've seen the procedure, I'm going to go ahead and turn off the video here, and I will post a picture at the end to show you what it looked like at the end. But we can clearly see, if I rotate it so you can clearly see better, that we do have the pigment separating out, that we do have a lighter green color here at the top. We have a darker green underneath that. So our spinach, we can already tell, has multiple colors of pigment. So we do see, based on our diagram, that chlorophyll A is a bigger molecule and does not travel as far as what we would expect for chlorophyll B, and that the yellowish colored pigments, the xanthophylls and carotenoids, the yellow-orange pigments, are our lightest weight molecule, and they should travel the furthest. So again, once this has migrated upward, I will take a picture and share that with you as well as a ruler, so you can actually get some measurements as to how far the different pigments have moved. With that, have a great day, and I'll be back with more kitchen science lab later on. Take care.